you hear me? Okay. So thanks to you for the nice introduction and so how embarrassing introduction. And so thanks again for coming. So today I'll talk about the mechanisms of memory shape transitions. The reason we are interested in memory shape is very straightforward because when you look into a cell, you see all these memory bond structures, and they have a wide range of different shapes. And usually we talk about membrane shape in terms of membrane curvature, which means for the cell membrane, we have high curvature regions such as the trafficking vesicles or membrane tubules, and we also have low curvature regions such as the plasma membrane. And it's crucial for the cell to maintain this highly curved structure in order for the cell to be alive, also for the cell to communicate with each other. So today we are going to focus on the plasma membrane and talk about how external materials may get into a cell through a process called endocytosis. But the principles really apply to all situations where you have membrane shape transition. So here is the 10 times closer view of the classical endocytosis. So as you can see, a number of different proteins are involved in this process. And among these proteins, a major family is called borrowing proteins, which means if you look another 10 times closer, you'll see that the membrane binding interface of this protein has this intrinsically curved structure. So this, as you can imagine, allows the protein to stabilize or generate high membrane curvature. So for the classical endocytosis, different proteins are sequentially recruited to the plasma membrane, and there are many different borrowing proteins working together in order to achieve this membrane deformation process. So, however, we know that this is not the only type of endocytosis. In fact, just earlier this year, people discovered the so-called endothelium-mediated endocytosis. So in this case, only one of these borrowing proteins, called endothelium, play a key role in deforming the membrane. And comparing different types of endocytosis, another major difference is in terms of their speed. So for the classical endocytosis, the rate is limited by the sequential protein binding process. So it takes about 50 seconds to go from a flat membrane to a highly curved vesicle. But other types of endocytosis can go much faster. So the extreme example is the so-called ultra-fast endocytosis, where endothelial vesicles are, so people found this in neurons, and the endothelial vesicles were observed within 50 milliseconds after the synchronized fusion of exocytosis. So the fusion of vesicles to plasma membrane will increase membrane area, therefore decrease membrane tension. So it was hypothesized that this tension decrease is the reason that travels along plasma membrane and trigger endocytosis at totally different location. So in our study, our goal is to try to understand this membrane deformation process. And especially we are interested in how mechanical properties of the membrane, as well as chemical properties of the protein, how those influence this membrane shape transition. So, but before getting into the experiments, I really need to mention that the reason this is still not well studied, because a major reason is because this deformation is really tiny. As you can see in this EM image, these membrane tubes produced by borrowing proteins have a typical radius of only 10 nanometers, which means if you look at these tubes under an optical microscope, it will just be a blur, and there's no way you can quantify the initiation of this membrane deformation. So in order to get around this problem, we were thinking if we have a cell-sized vesicle and put the vesicle into a solution, a bulk solution of borrowing proteins, and most likely the deformation will not happen just at one location. So indeed, as you can see in this picture, these blurry tubes are everywhere surrounding the vesicle. So this tells us, instead of trying to see individual tubes, what we really need to focus on is to look at the remaining membrane in the vesicle, because any change in this represents the contribution from all these tubes, which may be large enough for us to see. So with that in mind, we came up with this vesicle transfer assay, where we have two droplets of solutions. The green one represents a solution of protein labeled with a green dye, and the red one represents a solution of these cell-sized vesicles containing a red dye. And we can pick up a vesicle and move the vesicle into the protein solution. And by doing that, we can measure the protein membrane binding process. And the vesicles are controlled by this micrometer-sized glass pipette, which gives the vesicle a well-defined geometry of a spherical part outside the pipette and the cylindrical part sucked into the pipette. And by changing the aspiration pressure or the suction pressure, we can apply a well-defined surface tension on the membrane body. So the take-home message really is there is a technique which allows us to measure 3 independent quantities: membrane protein density, vesicle geometry, and membrane tension, and we can measure them at the same time. So here is what happens in the experiment when we move a vesicle into a solution of endothelin. So these are time-dependent confocal images, where the green represents the signal from the protein, and the red represents the signal from the lipids. And as you can see, as time goes by, protein density on membrane increases, and eventually you see these blurry tubes emerging from the vesicle. And if you look at the lipid channel, what you see is there is a dramatic decrease of this aspirated part of the vesicle. So this tells us the formation of these tubes is consuming the excess area of the membrane that used to be stored in the vesicle. So we can further quantify this by measuring the time-dependent protein density and membrane area. And from this, I hope it's clear that what it shows is after the protein density reaches a certain level, membrane area begins to decrease. And from the starting point of area decrease, we can define a transition density, which essentially tells you how much protein is needed to deform a certain piece of membrane. Another thing interesting we found in this experiment is if we do the experiment under high membrane tension, this process will not happen anymore. So example of that is shown over here. As you see, a high tension vesicle is transferred into a solution of endothelin. Nothing really happens except for protein membrane binding. However, at this point, if you decrease membrane tension, again you see the tubes begin to emerge from the vesicle, and this sucked area disappears. And we know that this tension decrease can equilibrate across the whole cell-sized vesicle within milliseconds. So this provides the potential explanation for the ultra-fast endocytosis. Because if you remember, in this type of endocytosis, membrane deformations or endocytic vesicles were found within 50 milliseconds after the tension decrease caused by the fusion of exocytosis. So this also tells us this tubulation or this membrane deformation process is tension dependent. And we can further quantify this process by measuring the transition densities for vesicles under different membrane tensions. And eventually we get this stability diagram where each data point here represents an individual vesicle, and the black triangles represent the transition densities for vesicles of different tension, and the white circles represent the maximum density of a vesicle that didn't show tubulation. So as you can see, this tells us there are 2 states of the membrane, a planar or stable state, and an unstable state with these membrane deformations. And there are 2 ways to go from one state to another. One is by increasing protein density on the membrane, which is a rather slow process, and the other is by decreasing membrane tension, which can be much faster. And we get this stability diagram for a certain lipid composition mimicking the inner leaflet of plasma membrane where endocytosis happens. However, we wonder if the lipids is actually influencing the position of this boundary. So, as you may know, a key feature that defines different types of lipids is the shape of the lipids, which means we either have these conical shaped lipids, such as DOPE, or we have these cylindrical lipids, such as DOPC. And the way people quantify this is in terms of lipid spontaneous curvature, which means if in this case, a more negative value means a more conical shape. And in our experiment, we can simply replace the conical DOPC with a bunch of different, the cylindrical DOPC with a bunch of different conical lipids. And what we found was, in the presence of conical lipids, much less endothelin is required to deform the membrane. And so this tells us the presence of conical lipids in the membrane will serve as a third level to control membrane deformation. And following that, if we add this anti-conical lipids into the membrane, the transition density will be increased. And by changing the fraction of this anti-conical PIP2, we can even give an estimation about the unknown lipid spontaneous curvature of PIP2, which is roughly 1/3 per nanometer, representing an inverted DOPE shape. And if you're familiar with lipids, you know there's another, in addition to lipid shape, another feature that defines different types of lipids is the charge of lipid head group. And in this case, we can roughly define lipids into neutron lipids such as PC or negatively charged lipids such as PS. And the charge of the membrane is crucial for the membrane binding of borrowing proteins. And in this experiment, we simply replaced DOPS with DOPC. And what we found was indeed with a higher percentage of charge, the protein binds stronger to the membrane, as indicated by an increase of equilibrium density. However, the transition densities, or the density required to deform the membrane, remains roughly the same for different percentage of charge. So this tells us the membrane binding kinetics controlled by the charge of the lipids is not really influencing the membrane deformation. And in order to understand this process, what we really have to consider is thermodynamics. So with that in mind, we can simply write down the thermodynamic free energy of the protein membrane system, where here we use H to represent the surface profile of the membrane, so that the Laplacian of H will be the local membrane curvature. So in the first part of this equation, talks about the elasticity of the membrane, with kappa representing membrane bending rigidity and sigma representing membrane tension. And the last part of the equation talks about the interaction between proteins on the membrane. The A term is the energy of having a protein density of phi, and the B term is the energy penalty for having an inhomogeneous protein distribution. And what's interesting is this middle term, which contains both protein density phi and also the membrane curvature. So this tells you that this represents a coupling between protein and membrane curvature. And with the coupling strength represented by C0, which is the spontaneous curvature of the membrane after protein binding. And we can go ahead and determine the thermodynamic stability of this free energy expression. And it turns out to be a simple three-term equation, with the first term talking about this coupling strength, and the second term containing sigma, which is membrane tension, obviously talks about the contribution from membrane tension. And the last term containing A can be related to protein density by treating the protein as a two-dimensional gas on the membrane with a certain attraction alpha and certain size beta. And so with this, we eventually have a fitting equation correlating membrane tension with protein density on the membrane. And in the equation, this bending rigidity we can measure, and the size of the protein we can estimate based on the crystal structure. So in the end, we only have three fitting parameters representing three unknown molecular properties of the system. So we can use this equation, fit A to our experimentally determined transition density. And what we found, so by fitting this, the three things we can learn about the three unknown parameters. The first one will be the spontaneous curvature of the membrane induced by the protein, which actually agrees pretty well with existing literature. And the other two parameters, as in this figure, are the protein density to tubulate a zero tension membrane and the upper tension limit where beyond which membrane will never be tubulated anymore. But you may wonder what's the meaning of all these numbers. Just to give you an example, resting the membrane tension and endophilic density for a resting neuron lies somewhere over here. So as you can see, the cell is pretty close to the transition boundary, which means you can easily go across the boundary by locally concentrating the protein or by changing its membrane tension. So a brief summary of the first part. So we started with cell-sized vesicle. A zoom in of the vesicle will look something like this, where we have a roughly flat membrane with some protein on the membrane, and the membrane is under a certain membrane tension represented by this weight. And now imagine you are the vesicle, and now you want to go through a membrane shape transition process. And since you heard about this presentation, hopefully you know that you should follow this membrane stability theory, which tells you there are three ways you can go. The first is to recruit more protein on the membrane. The second is to decrease the membrane tension or decrease the weight. And the third is to change the lipid composition or incorporate more of these conical lipids into the body. So now if you may change your perspective and imagine you are one of these borrowing proteins. So you are sitting on the membrane, and the question will be if there is anything you can do to facilitate this process, or if there is anything the protein can do to facilitate the membrane deformation. To